So yeah, first I would like to thank the organizers, the awesome JGI team, for selecting me to give a talk today. I've been coming to this meeting for many years now, and I always very much enjoy it. So today I'm going to talk about one of our newest method developments, which is a method that allows us to look at carbon flow in microbial communities. So, and this method is based on metaproteomics, so I'm going to give 2 slides to just very briefly introduce metaproteomics. So metaproteomics is an umbrella term for all approaches that allow us to identify and quantify proteins from uncultured environmental communities. So in that, it's really a natural extension of metagenomics, and we have heard a lot about metagenomics already at this meeting. So while metagenomics allows us to get the genomic potential for metabolism and physiology and interactions in microbial communities, metaproteomics then really allows us to figure out what of this potential is really being expressed, which metabolic pathways and which physiological activities are really of relevance in the environment. And I cannot go really into the details on how metaproteomics works. One of the important things to know for this talk is that most of the approaches that we use rely on high-end, high-resolution, accurate mass spectrometry. A question, or one thing that I wanted to briefly point out is, because it's often of interest, is how much material we need to do metaproteomics. Nowadays, the sample preparation methods have improved vastly over the last few years, and we can now work with very, very little material and oftentimes get very good metaproteomic coverage already with 0.5 milligram of material. So just to introduce briefly the breadth of questions that we can address with metaproteomics before coming to the actual new approach that we just developed. One of the most classical uses of metaproteomics, which has been around since 2004, we use it to understand the metabolism and physiology of microbes in communities, and we can answer questions such as what are the nutrient and energy sources of microbes in a community. We, for example, use this to show in a marine symbiotic worm that harbors symbionts, that these symbionts can use carbon monoxide as an energy source. We can also use metaproteomics to show how a microbial species interact. And recently, we also developed metaproteomics into a tool to study community composition. And we can now study community composition in terms of biomass and not only in count. So while most of the approaches that are being used, like such as RNA sequencing, really look at counts of microbes in communities, metaproteomics now allows us to look at the community composition in terms of mass contributions of individual members. So we get 2 different perspectives on communities. So one of the questions we couldn't answer until very recently is what did microbial species X have for breakfast? And we think we have now an approach that allows us to at least in part answer this question. And just to specify the problem that I want to address a little more, just imagine your favorite microbial community now. And let's just assume you have been spending a lot of time studying your community, right? You have been doing RNA sequencing, you have done metagenomics, you have done metatranscriptomics, metaproteomics, metabolomics, all these different approaches to really get at what the individual members in these community are doing. And from all this data that you acquired, you found out that one of these members, let's say this gray big bacterium here, has an important role in this community. Maybe it's a pathogen in a community, or maybe it's an important player in a biogeochemical cycle. You get this image. You learn a lot about the different pathways that play a role. And one of the things that seems to be really crucial for understanding this microbe is that it has an abundant sugar transporter. And you spend a lot of time and effort on trying to figure out what the annotations for the sugar transporter are. And you can really not get the substrate really nailed down. There's a lot of different sugars it may potentially be transporting. And from metabolomic data, for example, you might know that you have different sugars in this environment that are abundant, that may be the substrate of relevance. So let's say we have a sugar A, which comes from one source. It could be maybe from root exudates. But you have the same exact sugar coming from a different source in that environment. And then you also have a sugar B and C and D and so on and so forth. So now it's important for you to understand what is the actual substrate for this organism so that you can understand the interactions. And so if you actually knew isotopic signatures of the sugar and the microbe, then maybe you could link them. And so we are using an approach that is based on carbon isotope ratios. And so I'm going to give a brief refresher on carbon isotope ratios and how we can use them in biology to answer questions before I then describe the approach that we are using. So as you all know, for carbon, we have two different stable isotopes in the environment. We have carbon-12, which is the most dominant one, makes up almost 99% of all carbon on Earth. And then we also have carbon-13, which makes up roughly 1%. And when we look at the amount of carbon-13 we have in any kind of material, we always express this as a comparison to a standard. So we express this as this delta carbon-13. So if we have a material, we compare it to the standard. If the material has more carbon-13 than the standard material, we call it heavier, and it has a positive value. And if it has less carbon-13 than the standard, we call it lighter, and it has a negative value. So just to briefly introduce the two main questions where carbon isotope ratios have been used in the past to answer questions in biology, one of the main questions that we can answer is, what is the food source of an organism? And this question can be answered based on the paradigm that you are what you eat. So you should have the same carbon isotopic signature as your food source. And just to give you a brief example how people have used it in the past, fortunately, Victoria Orphan already introduced the anaerobic oxidation of methane in her excellent talk on Wednesday, so I don't have to introduce that. So one of the things, when Victoria started out with her research quite a while ago, they didn't really know who is responsible for consuming the methane in these sulfate-methane transition zones. And so what they did, they used a mass spectrometry imaging method, and what they found is that when they imaged these consortia, that they found that the archaea in these consortia had a very light isotopic signature that corresponded to the isotopically very light methane in the seafloor. And so they could really make the connection there between the methane and the archaea that actually consume the methane. Another way we can use carbon isotope ratios or stable carbon isotope fingerprinting is we can actually answer the question sometimes which pathway was used for carbon assimilation. And this is now based on the paradigm and which is actually the opposite of the previous one. You're not always what you eat. Sometimes the pathway that is used for carbon fixation, especially carbon-1 compounds, actually changes the isotopic signature of an organism. So between what the input carbon and what is actually in the organism. And so if we look at these different carbon fixation pathways, each one of them has a very specific signature that it introduces, a very specific change in the carbon isotope ratio when a particular pathway is used. So now, the standard method to measure carbon isotope ratios and other isotope ratios is isotope ratio mass spectrometry. And what you usually do with this approach is you just burn up your sample and you measure it. So what would happen if you do isotope ratio mass spectrometry on your microbial community sample, instead of getting all these nice values, very informative for each of the members in the microbial community, you get this bulk value that just gives you an average, and it doesn't really tell you about what the individual members of your community do. So that's what we wanted to address. We wanted to see if we can extract stable carbon isotope fingerprints for individual species from our metaproteomic data, which is high-resolution mass spec data, so it has all the information we would need. And we termed this approach direct Protein-SIF. So why we thought we would be able to do this is the following. So if we look at one of these spectra that we get to identify peptides and proteins, and we get thousands to 100,000 of these spectra in each metaproteomic run, we identify the peptides, and let's just say, we can say we get a specific peptide sequence, and we know it's identified from a specific organism. So we can make the connection to the taxonomy. So when we look at one of these spectra, we see all these different peaks. All of these peaks correspond to a single peptide. So it's quite complex spectra. And now if we zoom in just into one of these peaks, we can actually see that one peptide actually composes multiple isotopologue peaks. So the first peak here is what we call the monoisotopic peak, where all the atoms in the peptide have the normal standard stable elements. And then in the second peak, one of the atoms in the peptide is actually exchanged for one of the heavier or stable isotopes, for example, carbon-13 or N15 or something. And then the third peak has two of them, and so on and so forth. So we get really nice isotopic patterns that we can extract from our data, and we can link them to the taxonomy based on our identifications. So to make a long story short, we spent quite a while developing this together. I developed this together with Mark Strauss and Jolie Dong, my colleagues from my previous university, the University of Calgary. And what we did is we developed an algorithm that allows us to extract these isotopic patterns. We then simulate in silico all kinds of different isotopic patterns on an incremental range. We compare them to the experimental patterns, and then we find the best fit, and we determine with the isotope ratio for a specific peptide. I really love preprints, so if you're actually interested in the method, you can already read all about it, despite the fact that it's still in revision. Yes, so we developed this approach, and then of course we did some proof of principle. We had to see if it actually really works. So what we did, we used pure cultures of bacteria and archaea, which we then could of course measure with the isotope ratio method, isotope ratio mass spectrometer, which is the gold standard method, and then with our own approach. And we then can compare both measurements against each other, and you see we get a very nice correlation of the two values. So the method really works. The next thing, totally forgot about that. And one thing we see is that, of course, autotrophic organisms have a lighter signature than the heterotrophs. We also figured out that this particular cow that donated the BSA that we use as one of the reference calibration standards must have been eating a C3 plant, and that the one human that we measured also fits very nicely into the picture that we know from the literature. We actually, one of the things I'm not going into much detail today is we actually need a calibration standard for the method, and we determined that human hair is an excellent calibration standard for the method. So the next thing we did is we mixed, from our pure culture organisms, we mixed different kinds of mock communities where we had different ratios just to see if it also works in community samples. And the main point of this particular slide is now, so on the y-axis we now have the IRMS measurements as compared to our Protein-SIF measurements. And we can see, and then we have different window sizes, different numbers of peptides that were measured per species. Each of the dot is a measurement for one species. And you can see that the more peptides you have, the more accurate the measurement gets. So this is an important point. You really need to get a good number of peptides for your species to get an accurate measurement. So just to give one brief case study how we can apply this particular method and what we can learn from it. Here we have the gutless oligochaete Olavis algarensis, which lives in these nice sandy sediments in the Mediterranean. And the important thing to know about Olavis algarensis is it has no digestive system, so it cannot eat any food. Instead, it has a very thick layer that you see here now in the cross-section in red and green. And this thick layer is a symbiont layer. So there's tons and tons of symbionts that take care of the nutrition of the worm. So we had done many prior studies. There's a famous, one of the first really good metagenomics studies that were ever done by Tanja Woike. It was published in 2006, and we had a metaproteomics study from both these studies, we learned a lot about the physiology and metabolism of this particular symbiosis. What it boils down to in terms of what we thought after doing these studies in terms of carbon metabolism is we have these two green oh, sorry for the people that are red-green blind. I didn't think of that. Okay, should have thought of that. Well, anyway, we have some sulfur oxidizing symbionts that fix carbon dioxide. And then, so this is the initial step where the carbon comes into the holobiont, and then everything else supposedly relies on this carbon. So we have the carbon is transferred to the host, as well as then in form of waste products to the other symbionts. And then we also had a spirochetal symbiont where we actually didn't know what it's really doing because we didn't have enough data at the time. So our expectation really was that all the carbon is derived from this initial fixation event. So everybody should have the same signature, basically. And okay. So yes, so that was the expectation. We had bulk measurements of the worm, so it was -30. So our expectation was that everything would be in the range of -25 to -35. And then we did the Protein-SIF. And fortunately, most of our prediction came true. Most of the symbionts actually, and the host as well, were in the range of the prediction. We had -25 for the host, -32 for some of the symbionts. But then we also discovered that there was one big outlier where we have a symbiont for which we predicted it should be deriving carbon symbiosis internally, but it actually had a much, much heavier signature. So we're currently scratching our head, and we have to go back and really better understand how this works. Yes, so in summary, There's many different questions that you can address with metaproteomics. We developed this Protein-SIF approach that now allows us to look at the food source and carbon fixation pathways for species in microbial communities. And with that, I would like to thank you for your attention and the many different people that were involved in this work. I want to give a shout out again to the JGI, because without genomes and metagenomes, all the metaproteomic work that we do would be utterly useless, because it really relies heavily on having good, well-annotated metagenomes and genomes. And I also wanted to briefly highlight that I'm currently looking for postdocs, so if you're interested, just check out the opportunities on the website.
Thank you very much.